This is lecture 5B, part 1. We're continuing with cardiovascular physiology, the vascular part of it. The control of blood pressure is extremely important in maintaining appropriate cardiovascular function and adequate perfusion of all the different tissues. Now, the regulation of it or control of blood pressure involves two different main groups of control mechanisms. One is short-term and the other are long-term controls. The short-term controls include neural control through the nervous system, and the other is through blood-borne chemical compounds, which I'll discuss. These short-term controls will counteract any changes or fluctuations that occur moment to moment in the blood pressure, and they do that by altering overall peripheral resistance. That means the resistance to flow in the systemic circulation. We're going to go through these one at a time. The first short-term control I'll talk about is regulatory mechanism. I'll talk about are the neural controls. These neural controls act on peripheral resistance by altering the blood distribution depending upon the demand. So depending upon what is required physiologically, then the demand, the need at the moment, then it can alter the distribution. They also can maintain the mean arterial pressure, overall mean arterial pressure, by altering the blood vessel diameter and therefore changing resistance. These kind of neural controls operate via reflex arcs. So we're talking about autonomic reflexes, and the arc we're talking about, a reflex arc involves sensory tissue sending signals into the central nervous system where processing occurs, and then a signal goes out, a motor signal goes out to effector tissue, which in this case is smooth muscle tissue. Now, in this case, the sensory tissue that pick up the important sensory information are the baroreceptors. And as you know, you have baroreceptors, pressure receptors, in other words, in your aorta and the aortic body in the aorta, and in the two carotid arteries, you have baroreceptors in the carotid bodies. These send information to be processed in the vasomotor centers of the brainstem in the medulla, and then will send vasomotor signals out to the effector tissue, which is, if needed, they'll send signals out from the medulla of the brainstem to the vascular smooth muscle. So that's the entire reflex arc. Okay, so it's going from sensory tissue into the medulla of the central nervous system where it's processed, and then if needed, motor signals are sent out to the vascular smooth muscle effector tissue. The vasomotor center in the medulla is a cluster of sympathetic neurons that can control blood vessel diameter. They can send out signals that will control the blood vessel diameter. They also maintain overall blood vessel tone by the fact that they innervate the smooth muscle of the blood vessels, especially the arterioles, where the most control over diameter occur, the most main changes in diameter occur. It's important to have blood vessel tone so that the blood vessel is not completely relaxed and therefore has the maximum diameter. the reason that's important is because if you have tone, in other words, if you have a certain background level of output so that the blood vessels are at least somewhat vasoconstricted, the smooth muscle is somewhat contracted, then in that case there are two possible options. One is that the blood vessel could further constrict, or it could vasodilate. Okay, let me make it bigger. We could get vasoconstriction or vasodilation. However, if it's completely relaxed, then the only option is one, and that's vasoconstriction. You cannot get vasodilation. And so blood vessel tone is quite important. So there's a constant background level of signals going to the smooth muscle of the arterioles and, well, overall circulation, but particularly the arterioles. And in that way, you can have these two possible outcomes if need be. There's also a cardiovascular center, and there's a cardiovascular center which is made up of the vasomotor center plus the cardiac centers. The purpose of the cardiovascular center in the medulla is to integrate blood pressure control by changing cardiac output and blood vessel diameter. So in this way, there's overall control of the cardiovascular function, so it's appropriate in different situations. The vasomotor activity, if it involves sympathetic output, if it's increased, then that results in vasoconstriction, and we're talking about generalized vasoconstriction to get an overall rise in blood pressure. If you get systemic vasoconstriction, you're going to get a change in the space available for the volume of blood the person has, and therefore that's going to raise up the blood pressure. If it decreases, in other words, if sympathetic activity decreases from vasomotor centers, then you'll get vasodilation. It's I'm not sure why it's not here, but it should say, it should point out that this results in vasodilation, and the blood pressure accordingly will decline. It'll be reduced. Vasomotor activity can be modified by sensory information picked up by the baroreceptors and also chemoreceptors that are found in the same aortic bodies and carotid bodies, which are sensitive to PO2, that's the partial pressure of oxygen, PCO2, partial pressure of carbon dioxide, and also pH. It can also be modified by higher brain centers. Whenever you see this term relative to what you're talking about, it means a more complex level of control. And for the vasomotor activity, which is found in the brainstem, that higher brain centers means output coming in from the cerebrum, so the cerebral cortex. The other thing that can change vasomotor activity are blood-borne chemicals and also hormones. So you can get baroreceptor, as I said, can affect vasomotor activity. So baroreceptor initiated responses or reflex responses. In this case, the initial information is an increase in blood pressure. That's the stimulus. The result is that information goes into the cardioinhibitory center, And the output is vasodilation, an increase in vessel diameter, decrease in heart rate, therefore a decrease in cardiac output, and of course a decrease in peripheral resistance because of the increase in vessel diameter. End result, a decrease in the blood pressure, which is the appropriate response, seeing as though the original stimulus was an increasing blood pressure. And when we say increasing blood pressure, it means above what's required in that situation at that time, and therefore all these mechanisms kick in, all short-term mechanisms, and the blood pressure is corrected down to an appropriate lower level. If the baroreceptor initiated reflex is initiated by a drop in blood pressure, declining blood pressure, if that's the original stimulus, then that goes to the information goes into the brainstem, and the processing is carried out by the cardioacceleratory center, and the output is an increase in cardiac output. It goes into the cardioacceleratory center, and the signals that go out, the effector signals that go out, result in an increase in cardiac output and an increase in peripheral resistance. As a result, and the other thing is that the low blood pressure also stimulates the vasomotor center to cause vasoconstriction, generalized vasoconstriction. Actually, that's increase in peripheral resistance covers that. And then, of course, the end effect, by increasing cardiac output, by increasing peripheral resistance, there's an increase in blood pressure. So that original stimulus is respond, the information is processed, and the response, the end result is an increase in blood pressure back. Again, short-term mechanisms. These are baroreceptor initiated reflexes. And this is outlined here on this diagram. Everything I talked about here, where the stimulus is a drop in blood pressure, all the events that occur which lead to an increase in blood pressure back to the normal appropriate range. And if the stimulus is a rise in blood pressure above normal or what's appropriate, then these responses occur and it goes back to the normal level. And you should go through this diagram. It's a useful diagram to make sure that this is clear, it makes sense to you, and it's, you would be able to describe these kind of events occurring in these situations where you have a response triggered by a short-term baroreceptor initiator response because of a rise in blood pressure that needs to be corrected, or a drop in blood pressure that needs to be corrected. Okay, the short-term mechanisms that involve chemical controls are chemoreceptor reflexes that initiated by responses to changes in PO2 and PCO2. So the baroreceptors respond to pressure, and chemoreceptors are responding to PO2 and PCO2. These chemoreceptors are found, as I said, in the carotid bodies and in the aortic bodies. And these reflexes can regulate the blood pressure because the sensory information is sent into the brainstem, into the medulla, and the information is integrated and processed. And also higher brain centers can modify BP, blood pressure, by sending information to the medulla as well. And in response, there's signals sent out to the effector tissue, the appropriate effector tissue, to respond accordingly. So, the chemical hormones or molecules that can result in a release of them into the bloodstream that can result in increase in blood pressure include release of norepinephrine and epinephrine. And in this case, we're talking about hormone release into the bloodstream, not neurotransmitter release at the end of postganglionic neurons. So we're talking about signals that are released from the adrenal medulla. The adrenal glands, as I've told you, if you look at the kidney, there's a kidney. The adrenal glands sit on top of the kidney, on each side of the both kidneys, and the medulla is the center part of the adrenal gland. The outer portion of the adrenal gland is called the cortex, adrenal cortex. This is the adrenal medulla. If it gets a signal from the sympathetic nervous system, sympathetic division of the autonomic nervous system, then it will release these neurotransmitters, and they will be released into the bloodstream and will result in an increase in blood pressure because of their effect on the heart. Another hormone that can cause a change in blood pressure is antidiuretic hormone. Antidiuretic hormone on its own can cause an intense vasoconstriction. This is a systemic generalized vasoconstriction, particularly in arterioles, and this is triggered when there's a very low BP, which can result in release of antidiuretic hormone. The hormone itself is produced in the hypothalamus within the brain, and then it's released and it travels down to the posterior lobe of the pituitary gland. The hypothalamus has a stalk which extends down to join the pituitary gland, and this hormone, ADH, or antidiuretic hormone, is stored and, when needed, released from the posterior lobe. It is also known, and you'll see this quite often in literature and textbooks, it's an older name, but it's also called vasopressin. Antidiuretic hormone not only affects the arterioles, but it also has a strong effect on the kidneys that we'll talk about in later lectures. Another hormone that can increase, and by the way, another hormone that can be released and cause an increase in blood pressure is called angiotensin II. It can also cause intense vasoconstriction. The kidneys will release, under certain circumstances, when actually blood pressure in the kidney is low, they will release a hormone or molecule called renin. Now, renin has enzymatic activity. It will act on a protein molecule that's in the blood plasma called angiotensinogen. By now, I would hope that you know that if the name of a protein ends in OGEN, that means it's inactive. But what renin does is it will activate angiotensinogen to form angiotensin I. It does that by cutting or cleaving the protein so that the protein is now active angiotensin I. So now in your plasma you have angiotensin I, and when it gets to the lungs, it gets converted to angiotensin II. So in this case, renin has enzymatic activity which activates angiotensinogen by cleaving it to form angiotensin I. Once angiotensin I is formed, it has enzymatic activity which can renin has enzymatic activity on angiotensinogen. It can cleave it to form angiotensin I. And then angiotensin I is converted to angiotensin II by an enzyme in the lungs called angiotensin converting enzyme, usually called ACE, ACE. So the angiotensinogens in your blood plasma, it's it's circulating. If renin gets released, it cuts it to form angiotensin 1, which then gets converted to angiotensin 2 by ACE when that gets into the lungs. The angiotensin 2 has a number of different important effects, including intense vasoconstriction. It has other activities which can increase blood pressure, and we'll talk about those later. I'm not sure where, but we will talk about it. Compounds that can decrease blood pressure. These are, again, short-term controls. Release compounds or chemical compounds that can reduce blood pressure if it's high. One of them is called atrial natriuretic peptide, or ANP. You may see it also called sometimes atrial natriuretic factor, but the more appropriate up-to-date name would be atrial natriuretic peptide. It's a protein that's found in the wall of the atrium of the heart, and when the blood pressure is high, that stretches the heart more. There's more blood getting into the heart. With the higher blood pressure, you get stretching of the heart, and that causes release of atrial natriuretic peptide. This peptide has a number of different effects, but overall, the general bottom line is that it will cause a drop in blood pressure and it'll cause a reduction in blood volume and blood pressure. It drops blood volume by triggering sodium and water release, increase in sodium and water release in the kidneys. So you excrete more sodium and water. Water goes along with the sodium. The ANP the natriuretic part of the name refers to the fact that sodium, the symbol for sodium is Na, and Na stands actually not just for sodium, but actually for an alternate name, which is natrium. So Na is the symbol for sodium and natrium. In many countries, sodium is not called sodium, it's called natrium. And this causes natriuresis, which is release or excretion of sodium in the kidneys. Wherever the sodium goes, water will follow, and you get a drop in blood pressure, and as a result also in blood volume, and therefore also a drop in blood pressure. Another chemical compound, which I won't discuss in any great detail, is actually a gas which is dissolved into the blood plasma called nitric oxide. It has very brief, short-term, localized, but very potent vasodilating effect. Other things that can cause vasodilation, and I would learn this one certainly, is histamine, but there are other molecules that are potent vasodilators. Another vasodilator which is not produced as a compound in the body but is often ingested is alcohol. Alcohol will cause a drop in BP because it inhibits antidiuretic hormone. Antidiuretic hormone, I would remind you, inhibits, and this is the anti part, it inhibits diuresis. So diuresis is the release of water, excretion of water by the kidneys. So this antidiuretic hormone being inhibited will cause an increase in water excretion by the kidneys, which is why people pee more when they drink alcohol, because of this inhibitory effect on ADH. Okay, now let's talk about the long-term mechanisms. There's some overlap, somewhat, you'll see, but I mean some similarities, but these are longer-term mechanisms. long-term mechanism on pressure involving the kidneys is renal regulation, control of blood pressure by altering blood volume. If the stimulus is an increase in blood pressure, the response is that the kidneys will eliminate more water, and you'll excrete more water, so you'll form a more dilute urine. Overall, in effect, is a reduced blood pressure. This takes some time, not a huge amount of time, but certainly is much slower than some of the other mechanisms I talked about. Similarly, but the inverse, if the stimulus is a decrease in blood pressure, the response is the kidneys will increase blood volume. They don't so much increase as help maintain the blood volume and reduce their loss of water. The overall effect is an increase in blood pressure. Unfortunately, people with chronic high or chronic low blood pressure, eventually the baroreceptors adapt to that and stop responding. So they won't respond, they won't send signals into the brainstem, and you won't get these kind of long-term mechanism effects. Kidneys act directly and also indirectly in maintaining long-term blood pressure regulation. Direct renal mechanisms involves alteration in blood volume, and indirect mechanisms involve the renin-angiotensin system, part of which I described. I described just one effect of the renin-angiotensin system, which is the activity of angiotensin II on the fact that it can cause vasoconstriction, but there's more to it, and I'll describe that here. Okay, we're going to talk about the renal function and regulation of blood pressure and long-term mechanisms of renal control. In the situation where you have a declining or drop in blood pressure, as I've told you before, renin will release, the kidneys will release renin. They're sensitive to the drop in blood pressure, and they'll release renin, which will act on the protein angiotensinogen, which is in the blood plasma, and that forms angiotensin I by an enzymatic cleavage of angiotensinogen to form angiotensin I. And then angiotensin I is converted to angiotensin II by ACE in the lungs. As I've said before, the immediate short-term response to that is that angiotensin II can act as a potent vasoconstrictor, but it also has longer-term effects in that it will stimulate aldosterone secretion. Aldosterone is produced in the adrenal glands, in the outer portion of the adrenals, which is called the adrenal cortex. So it's released from the adrenal cortex. I'm not going to write cortex here. Aldosterone enhances renal uptake or reabsorption of sodium, and therefore with renal uptake of sodium you'll also get water reabsorption. As long as there's ADH being released from the pituitary gland, it will also stimulate ADH release from the pituitary. And so you get increasing reabsorption of sodium and increasing water reabsorption from, the kidneys are constantly filtering the blood plasma and forming a filtrate, which goes through a tubular system, but most of that gets reabsorbed. But with aldosterone release, you get even a larger reabsorption. So you're going to be forming a very concentrated urine. You have to put out some urine to get rid of nitrogen waste, but this kind of situation where a drop in blood pressure is occurring, the kidneys are triggered to release renin that ends up producing angiotensin II, which does have effects that can raise blood pressure, but also directly, but also will act on the adrenal gland to cause it to release aldosterone. And aldosterone will increase sodium reabsorption, trigger ADH release to increase water reabsorption. These two typically always go hand in hand, water and sodium reabsorption or water and sodium release. And this is a flow diagram that goes through the kind of things that I've been talking about, all of the different types of controls, regulatory mechanisms involved in correcting a drop in blood pressure so that the end result is a return to an appropriate blood pressure for the situation. And you should go through this and make sure that it's clear. All right, let's move on. We can look at and assess cardiovascular function by taking the pulse and measuring the blood pressure. These are a couple of the vital signs. The difference, it's worth knowing the difference between a sign and a symptom. A sign, a vital sign, is something that has to be measured, whereas a symptom can be sensed by the person. But you cannot, you can sense if you have pain or nausea or something like that, but you cannot sense what your pulse or blood pressure are. So these are vital signs. They need to be measured in order to be determined, and pulse and blood pressure are two of them. The other two main ones typically are measured are respiratory rate and body temperature. The pulse is a pressure wave caused by expansion and recoil of elastic arteries, and it can be measured in a number of locations, but one of the easiest is the radial artery, so you can measure radial pulse, and it will change with depending on the person's health, their body position, and their level of activity that they've just recently, been carrying out. And this way you can have a feel, some, quantitative measure of the health of the cardiovascular system. Blood pressure is measured indirectly using sphygmomanometer. It's a pressure cuff placed on the upper arm, and we've gone through this in the labs, but I'll just quickly go through it. The pressure is increased until it's greater than the systolic blood pressure in the brachial artery, so that it cuts off blood flow and you no longer hear it. And then the pressure slowly gets released on that pressure cuff, and the person doing the measurement listens with a stethoscope for sounds at the place where the brachial artery comes down below the cuff at the elbow. And as soon as you start hearing sound, that's because blood is starting to seep through, as shown here. And so you'll hear the first of the Korotkov sounds when the pressure cuff hits just goes down below the systolic blood pressure. And then when you get below the diastolic pressure, then you have laminar flow of blood, and there's no sounds. There's no more, the Korotkov sounds stop. And in that way, it's quite easy or straightforward to, determine blood pressure. The first sound is the systolic. The second, not the second sound, but when the sound disappears, that's the diastolic. Blood pressure has a circadian rhythm. It'll cycle over a 24-hour period. Typically it peaks in the morning. There are many things that can cause variation in blood pressure. Age, mood, sex, weight, ethnicity. Sex is which one, not how much. Socioeconomic status has is a factor that can cause variation, physical activity, etc. So we describe hypotension as a low blood pressure in which the systolic is below 100. Hypertension is a sustained, continued elevated pressure where the systolic is above 190 and the diastolic is above, not 190, where the systolic is above 140 and the diastolic is above 90. There are constantly variations in your blood pressure depending upon posture, activity, time of day, but these transient changes are quite normal. It can be caused by many different things, but chronic elevation is not a good thing. Hypertension, chronic elevation resulting in hypertension is not good and is a major cause over time of heart failure, vascular disease, renal failure, and stroke. Hypotension can occur with body position, orthostatic hypotension, a temporarily low BP and dizziness when a person suddenly rises from a sitting or reclining position. We've spoken about orthostatic hypotension before. It can happen to even the healthiest young person if they've been lying flat for 8 hours and then they suddenly leap onto their feet. They can get a brief period of orthostatic hypotension, or they can even faint. Chronic hypotension suggests poor nutrition, could be a sign of Addison's disease. Which is an endocrine disorder I won't get into. Acute hypotension could be a sign of circulatory shock. Could be due to internal bleeding or sudden bleeding. You can get acute hypotension, a drop in blood pressure. It's can be a problem in patients who are undergoing surgery and intensive care. And those in intensive care always have some other problem going on. People in surgery, they're under anesthetic and unconscious and can lose proper control of their blood pressure and go into acute hypertension. All quite life-threatening, serious. Hypertension can be, again, transient, perfectly even appropriate. Sometimes if a person is suddenly intensely active, you get a big increase in the blood pressure. It can be, transient short-term, or it can be persistent, which is not good. It's described as primary hypertension or essential hypertension if it has a association with if it's due to effects that are associated with overall general health and that are affecting the person's overall general health. If it's secondary hypertension, it's because it's not it's due to some other identifiable and often, not always, often enough treatable disorder. Essential hypertension, on the other hand, is associated with certain risk factors. Primary hypertension risk factors include diet, obesity, age, which if you live long enough you cannot avoid, heredity, stress, smoking. Smoking is a really common cause of primary or essential hypertension. Sometimes hypertension, high blood pressure, is due to some other disorder, excessive renin secretion, for example, some sort of endocrine disorder, arteriosclerosis, atherosclerosis, or arteriosclerosis is not a reversible condition, and it is associated with progressive age. As a person gets older, they get changes in their vascular system, which can lead to secondary hypertension. We're going to talk about the flow of blood through tissues, and we'll talk after a little bit about changes in flow into different tissues like the muscle or skin or brain or lungs. We'll go through a number of different tissues where blood flow can change depending upon the conditions that the person's experiencing. But I think I'll stop here with Unit 5B, Part 1.